Have you ever wondered how scientists extract DNA from human blood cells? It seems like a process straight out of a science fiction novel, yet it's a routine procedure in many laboratories worldwide. Today, we dive into this intriguing world of DNA extraction. First, it's necessary to understand that the extraction process is rooted in a simple yet profound concept. DNA, or deoxyribonucleic acid, is the blueprint of life. Held within every cell of our bodies, it carries the instructions for making all the proteins that allow organisms to live, grow, and reproduce. But to study this DNA, scientists must first extract it from cells. The process begins with the collection of a blood sample, typically from a vein in the arm. This sample is then mixed with a solution that breaks down the cell membranes, releasing the DNA within. This solution, known as a lysis buffer, is composed of water, salts, and detergents. It's a bit like popping a balloon to get at the air inside. Next, proteins and other cellular components must be separated from the DNA. This is achieved by adding a protease, an enzyme that breaks down proteins. Once the proteins are degraded, the solution is subjected to centrifugation, a process that uses centrifugal force to separate substances of different densities. The DNA, being less dense than the other components, will float to the top of the solution. The DNA is then carefully drawn off the top, leaving behind the heavier proteins and cellular debris. At this stage, the DNA is still mixed with other components, so it's washed with an alcohol-based solution. This causes the DNA to precipitate or come out of solution, making it easier to see and collect. Finally, the DNA is dissolved in a small volume of a special buffer or water, ready for further analysis or manipulation. The DNA is now pure and can be used for a variety of applications, including genetic testing, research, and even forensic investigations. To summarize, DNA extraction from human blood cells involves several key steps. First, the blood cells are broken open with a lysis buffer to release the DNA. Next, proteins are broken down with a protease and then separated from the DNA through centrifugation. The DNA is then precipitated with an alcohol wash before being dissolved in a buffer or water for further use. This process, while seemingly complex, is a testament to our growing understanding of the microscopic world that exists within us. It's a fascinating journey, from a simple blood draw to a purified DNA sample, ready for the myriad of uses that continue to revolutionize our understanding of life itself.